everyone. I'm a staff scientist from ABPDU. Today I'll give you some summary of what we did about waste streams conversion. So I'll start with an introduction on myself. So I was born and raised in China. I got my bachelor's degree in chemical engineering. And then after that, I went to Institute of Process Engineering, Chinese Academy of Sciences for my master's degree. That was mainly focusing on physical chemical properties of analytic liquids, basically collecting data from the literature about the different properties and trying to link the liquid structure with those properties and predict the physical properties such as melting point. Recently, I learned that's actually called machine learning. And then I come to US for my PhD. My PhD work was on biomass fractionation using liquid-based process. So at University of Alabama, I worked with Dr. Rogers' group on this topic. Basically, we try to fractionate lignin from cellulose, hemicellulose instead of conversion. And then use them as a biopolymer to make biocomposites. In 2011, I joined JBay as a postdoc researcher. I think many of you on the same boat. And I joined a deconstruction division focusing on biomass to sugar and sugar extraction. In 2015, from 2015, I moved downstairs to work with ABPDU, especially in catalysis, recovery, and analytical team on the relevant projects. One of my research focus is on waste conversion. So this is the focus I'll discuss for the seminar. So just to introduce the feedstock I have been working on. So it's, as you know, we have those three bins generally in our household. Black bin for landfill, blue bin for recyclables, green bin for composable. So my focus is only on the landfill designated municipal solid waste. Basically, based on this EPA report, you can see still considerable amount of the material is organics, including paperboard, trimmings, and food, which account for almost 50%. So we have partnered with both national labs and industry for organics conversion. Another major component is plastics, which is accountable for 20%. And we are currently working with a startup company in Bay Area called X, and also JBay researchers on this topic. So I'll go through these two categories in the next session. So let's take a look about conversion of organics. I have 3 case studies here. First one is about MSW blends conversion, which starts actually from JBay when I was a postdoc here. And then conversion on landfill designated organic streams. That's a partner with local waste management company Ecology. And last one is upgrading our post-consumer solvent hygiene products. All right, many years ago, we started this project with Sandia National Lab and Idaho National Lab to look at MSW blends. So we choose here you can see this is non-recyclable paper procured from a waste management facility. Basically, it's from the landfill waste, and also yard trimmings. As you can see here, still a significant of yard trimmings in the landfill waste streams. So Idaho National Lab, they procure those feedstock and they did blends with our model feedstock, which is corn stover and switchgrass. And they also did the cost analysis. As you can see here, with blending of MSW, we have much more regions which can provide the feedstock with lower cost. So we want to evaluate how does that impact on the conversion side. At the beginning, we apply the liquid derived based acidolysis process. developed here from JBay. So basically, we do pretreatment using imine acetate, and then afterwards we lower down the temperature and directly add acid, which hydrochloric acid, to start the hydrolysis process. And basically, this is enzyme-free to use acid instead of enzyme for the carbohydrate to sugar conversion. And then we got this hydrolysis and solid residue. We have tried different blends, totally 16 MSW blends, also with different pretreatment temperature and other conditions. We basically optimize that process for MSW blends. Eventually we got around 80% sugar yield, slightly lower than enzyme, but the advantage is to use cheap acid instead of expensive enzyme. And then at that time I work with ABPDU on this scaling up of this process. Using 10-liter power reactor, we successfully scaled up this process and published a few papers. And later on, during that project, after I moved to ABPDU, we continued working on this project, and we applied the choline lysate-based conversion process, which is using choline lysate water to convert the biomass to sugars, and then use engineered E. coli stream developed also by JBay researchers to convert the hydrolysate to mesoketone, medium chain length mesoketones, which is a diesel precursor and also can be directly used as a blend stock. So this study tells us basically MSW didn't inhibit the conversion process, like compare it with the control feedstock. And this demonstrates the potential of using the low cost or negative cost of MSW, because in the waste management, if you go landfill, there will be involved in tipping fee. So in some cases, we can consider this as a negative cost waste streams. So to use that as a blending agent to supplement the standard biomass and reduce the feedstock cost. So this basically, in the future biorefinery, I think, because after those years and right now, MSW start to attract more attention, especially in vitro, for to be able to lower the feedstock cost in the bioconversion process. The next few slides is about the collaboration with Recology, a waste management company headquartered in San Francisco. They reached out to us because they read some papers about we published on the MSW blends. Collaboration with Father. It's an Italian company, a subsidiary of Procter & Gamble. For this project, we are evaluating the upgrading of post-consumer solvent hygiene product, which is actually dirty diapers. We developed this mechanical processing to separate cellulose-rich material from the solvent. Before sending us those streams, they actually already did plastic removal on the company side. If you are a parent, you know how much baby can generate dirty diapers on a daily basis. And when we visit Recology and also other waste management facility, they said diaper is a headache for waste management because they entangle. For example, the plastics and the material will kind of entangle with their meal or some equipment, which cause a lot of problems. So after this mechanical processing, we also did a biological conversion through enzymatic hydrolysis and fermentation to produce bioethanol as a proof of concept. A few years later, they were able to actually develop this pilot plant in Italy accommodating basically the mechanical processing, not the biological conversion, but mechanical processing. So in Italy, they set up this smart bins to collect diapers from consumer, use diaper from consumer, and then passing through their pilot plant. They were on a yearly, I think on a yearly basis, this is from their website, they collect 150 ton in that city of used diapers, and then they avoided 62 tons of CO2 and produced this much secondary feedstocks, including plastics, a solvent material, and cellulose. So cellulose can be used, like they use it to make books. So diaper book to books. So in next few couple of slides, I'll talk about the current ongoing project with X and JBIC on conversion of plastics. So I'll go through why we choose polyesters and the depromatization, biocompatibility, and TALC analysis. As you can see here, so plastics is everywhere. So in our daily life, during the past half century, plastic production has been dramatically increased. However, most of plastic has very low recycling rate. As shown in this figure, even for PET with the highest recycling rate is still only 18%. The majority of the plastic end up in landfill or leak to the nature environment, causing a lot of environmental issue. So we start with polyesters because these are relatively easier to break. We choose PET and PLA. PET, as I mentioned, is a major components in plastics, accountable for 50% of polyester. Only have below 20% recycling rate. During the current mechanical recycling, actually the PET lost molecular weight. Basically, so that's a downcycling process. So the quality of the recycled PET is not as good as the virgin resins. For PLA, it's the largest product sector of bioplastic. However, PLA can only be degradable under industry setting with high moisture and temperature. PET right now the production is small, but it has been kept growing. So PLA can be a contaminant to the PET streams. So we start with PLA and PET for ion liquid based depromatization process. So if you think about the polyester bonds in a water environment, the ester bonds can be broken either in acidic or alkaline conditions. So we choose 4 ion liquids, including both alkaline based, which is listed here, choline based, choline lysate, choline phosphate, and also 2 more imine acid, which is neutral, and imine chloride, which is acidic liquid, to evaluate the polyester depromatization efficiency. So we have on the left side, you can see the polymer depromatization. On the right side is the monomer yield. So we reached around 50%, 40% for both polyesters, and you can see the terephthalic acid yield is a little bit lower for PET. That's due to the oligomer formation during this process, which has been identified using GPC. So because the product is only analyzed using HPLC, so we cannot quantify those oligomer products. We also use simulation, computational methods to understand, I want to mention also here, we also compare it with water and with, because choline lysate is basic in 10% choline lysate in water. So we also using sodium hydroxide to adjust the pH to reach the same similar pH level to compare, but you can see clearly it's not just the pH. So we want to understand how the liquid interact with polyesters. So we use PLA as an example and perform the molecular dynamics. And here is some example of the results. This G value represents the the strength of the contact probability. Basically, the higher the G-value, that means it has higher contact interaction between the cation or the anion with the polyester ester bonds. So this is so the simulation results actually aligned with our experimental data, showing choline lysate has a stronger contact probability compared to other liquids. We move to the polyester mixture and using choline lysate in water. You can see with the increasing of the liquid loading, we got higher depolymerization or higher product yield. Because we all know we need to recycle. Generally, we need to recycle our liquid about 99% in order to make the economic viable. So we want to minimize the liquid usage. We're basically increasing the reaction time. So by doing that, we actually using 20% on the liquid, we were able to depolymerize about 95% of both polymers, and also get pretty high monomer yields. And so we got the depolymerized monomers. The next step is about to evaluate the bioconversion of the product, the monomers. So initially we use this Butita string, which can only catabolize lactic acid. As you can see here, the growth for lactic acid, depolymerized lactic acid, depolymerized PLA is similar to glucose, but for the depolymerized PET, it's mainly due to the presence of ionic liquid. Recently, we worked with JBay team on using this engineered string, Butita string, which can catabolize both PLA and PET, and we showed decent growth curve for both depolymerized PLA and PET. So this shows actually we can, so after depolymerization of plastic mixture, we can use both monomers as a carbon source for bioconversion. So using the aqueous biocompatible liquids allows us to direct utilize the depolymerized streams for bioconversion. And in the future, so this stream can produce PHA. So in our techno-economic modeling, we established this process to convert PET and PLA mixture to PHA, which is truly biodegradable plastics. So the process is like from MRF station, we will get the sorted PLA PET mixture, and through this one-pot conversion process for both catalysis and bioconversion, we produce PHA as a final product. So on the upper side, you can see the minimum selling price of PHA. In our current state of technology, it's still pretty high compared to the sugar-derived PHA. This is based on the current literature data and our preliminary catalysis data. However, if we, in the optimum case, if we assume the 90% conversion, especially 90% of bioconversion, and also other like higher solid loading and lower liquid loading, we can lower this cost, the PHA cost below around $1 per kg, which is much lower than the standard. Similarly, for GHG gas emission, the SOT is still pretty higher than the sugar-derived PHA. However, in the optimum case, we showed very promising GHG emission as well. Right now, we are working with X to do this high throughput screening of different liquids for PET depolymerization. Basically, we want to utilize X machine learning capabilities and to pass whatever commercial liquid we can get from the market into this pipeline and to identify, to train the model and to identify the optimum liquid structure, which can be depolymerize PET as a starting point. So right now we have about 100 liquids. Chang is leading this effort. So if you are interested in certain types of liquids, feel free to reach out to us. We have very small quantities right now, but we're happy to work with anyone who is interested. In the next couple of slides, I want to give you a brief summary about lignin depolymerization upgrading. I know JBay lignin depolymerization and conversion is also a focus of JBay, some of the JBay researchers. The first project is a part of the separation consortium working with Ariel to use membrane filtration to fractionate lignin, aqueous lignin streams. So in this project, we use sodium hydroxide and choline lysate as a catalyst to extract lignin from the cone stover, and then we pass aqueous streams through different molecular weight cutoff membranes. This cascading sequential TFF process, ranging from microfiltration, ultrafiltration, up to nanofiltration. Based on the GPC data, you can see TFF is capable of fractionating aqueous lignin streams. You can see the monomers has been enriched after passing through those membranes, and also the oligomers has been largely reduced. Also, the TA modeling showed a high initial concentration of the lignin streams play a critical role in the lignin, in low molecular weight lignin recovery. So recovery cost is much lower for choline lysate compared to sodium hydroxide because we have much higher lignin concentration in the feed stream. Another project also Chang led is a TCF project on lignin-derived liquids. So in this project, we start with using commercial lignin, trying to convert it to liquid. Basically, right now in the market, there's only a petroleum-based liquid commercially available. So we were trying to explore the feasibility of converting lignin to liquid through depolymerization, the intermediate separation and liquid synthesis. So we partner with Queen's University and a few companies, Ilium, Prionics, different roles. Basically, our approach is through lignin depolymerization, both oxidation and reduction. We generate the intermediate, including glycol and vanillin, and then we isolate those monomers for liquid synthesis. Then we evaluate the biopolymer processing and also have a TA and LC model. So briefly, for lignin oxidation, we use this alkaline condition with oxygen. And we applied a design experiment to identify the optimum conditions, which is listed here. Eventually, we got vanillin yield of 5%, which is pretty low. However, we were able to use ion exchange resin to be able to isolate this vanillin for further synthesis. The lower yield is mainly due to the craft lignin, as we know, it's the most challenging stream for conversion. However, we choose this stream because it's readily available, cost is relatively low, so it's more feasible as for a larger scale process. On the reduction side, we evaluate different solvent system as well as catalyst to produce this glycol and eugenol, those cresol as a product, phenolic compounds. So basically, we identify methanol as gives a higher yield, and also by tuning the catalyst, so we also can tune the product distribution for basically the chemistry. And the yield is similar to oxidation, is around 66% of the total glycol yield. So based on the preliminary data, we established this TA and LCA model, and you can see with a smaller scale current technology, the base technology, the cost is about $100 per kg, so which is pretty high. However, if we make it a larger scale production based on the lignin market, and also with optimum yield or increasing the yield of the precursors, we can lower down the cost to 14%, which is the ideal case. And similarly for GHG emission, we showed up to 85% reduction compared to the existing choline lysate in the ideal case. All right, that's all for what I want to share today. I would like to acknowledge all the team members, current and previous team members who has worked on this project. Also, we worked with a lot of JBA researchers. They have helped us tremendously on the analysis, catalytic conversion, and understanding of liquid chemistry. I would like to thank all the industry partners as well as national lab partners, as also for BDO for the co-founding of ABPDU. Thank you for your attention, and I can answer any questions you have.
A little bit of a grand picture one. How does the current pricing of this spout conversion compare to like other sources of fuel or materials? How much additional gap do we need to change to actually have market parity from an economic perspective?
Talking about in general, the waste conversion.
Yeah, for both of the kind of two waste conversion streams. Like, do we need like a factor of 2 increase in productivity to be economically competitive, or is it a factor of 10, or is it a factor of 100?
Okay, that's a great question. So I think for organics, we have pretty promising conversion yield based on my, experience working with paper and food waste. So both, sugar conversion and also bioconversion is comparable to I would say the other lignocellulosic biomass. It's comparable for plastics. There are a larger gap. So right now the conversion, maybe Chang knows more. So in our ideal case, we assume 90% yield, but the current even in literature, Chang, do you remember what's the conversion yield for PHA?
Like around
TPA.
Yeah, like 2 digits, like 10% off. It's pretty low, yeah. It's pretty low.
Yeah. So there are larger gap for plastic. If we want to do that, we achieve the bioconversion.
It sounds like the paper-based waste might actually scale economically and like essentially work out in the world to be more than just a research project, but actually something that like goes to gigaton or megaton scale, like worldwide, and plastics maybe less so.
Yeah, I would say paper waste just definitely easier.
I also had a question when you're talking about the waste streams. Just curious if you knew like how they sort the textiles from other organics, because then you said they hand sort food and plastic. That was really interesting.
Yeah. Because we went to Recology to see their sorting process. They have human sorting, so there's a conveyor belt. They just pass through everything, and then they are picking up like the fume plastic. Fumes plastic don't have a market, so have to be landfill. I showed the press. That's a pilot scale. It's not a commercial scale equipment. So that pilot scale, they can pass through everything, basically everything through that press. They based on the flowability, because the organics has very high moisture, so they tend to flow better. Also, they are softer compared to textile, metal. So based on flowability, they can separate those material from the others. But they also have, the very sophisticated process to sort out different types of plastics. They pick out that's in their material recycling facility, MRF facility for recyclables. But for the landfill, in commercial setting, they basically just send to for research. Yeah.
And film plastics, just any really thin plastic.
Yeah. Because that will entangle their downstream, their equipment, so they need to separate those out. All right. Thank you, everyone, again.